Good evening, everyone. Thank you for joining us tonight for this lecture with Suzanne Anker. Suzanne Anker is a visual artist and theorist working at the intersection of art and the biological sciences. Her practice investigates the ways in which nature is being altered in the 21st century, concerned with genetics, climate change, species extinction, and the non-human other. She calls attention to the beauty of life and the necessity for enlightened thinking about nature's tangled bank. She works in a variety of mediums ranging from digital sculpture and installation to large-scale photography to plants grown by LED lights. Her work has been shown both nationally and internationally in museums and galleries, including the Beijing Art and Technology Biennale at Cube, China, the Wuhan Biennial, China, the Everson Museum of Art, Syracuse, New York, the Daejeon Biennial, Korea, ZKM, Karlsruhe, Germany, the Walker Art Center in Minneapolis, the Smithsonian Institute in Washington, D.C., the Phillips Collection, PS1 Museum in New York, the J. Paul Getty Museum, the Para Museum in Istanbul, Turkey, the Museum of Modern Art in Kyoto, Japan, and the International Biennale of Contemporary Art of Cartagena des Indias in Colombia. Her work has been the subject of reviews and articles in the New York Times, Art Forum, Art in America, Flash Art, and Nature. She has hosted 20 episodes of the Bio Blurb Show, an internet radio program originally on WPS1 Art Radio in collaboration with MoMA in New York City, now archived on Elena Heiss's Clock Tower Productions. In November, Suzanne will receive the Lifetime Achievement Award from the Society of Literature, Science and the Arts, SLSA. The award recognizes a living individual for their exemplary interdisciplinary scholarship or artwork. In addition to all of that, there's something that I want to say because I have known Suzanne since 1993, when she came to the School of Visual Arts. Along with these accomplishments, she's a teacher and a department chair, and I think Suzanne exemplifies the type of seamless life where she manages to merge her teaching, her career, her studio practice, as well as her family life in a beautiful way that allows her to accomplish all of these extraordinary things. Congratulations, Suzanne. Thank you.
Thank you, everyone, for coming out tonight. This is my first lecture at home base. I usually travel around the world talking about these things, so it was very nice to see my family here. How did we sort of begin from this picture from the lab to the studio? Because I always say yes to everything. And in 2000, I think it was 2010, I got a call from David Rhodes. And he said, there's this building on 16th Street, and I offered it to the film department, but they didn't want it. So I said, I'll take it. And so what we did was spent the next few years fixing it up and integrating the traditional arts with new technologies. So that has been our mission since that time. The Bio Art Lab has been reviewed in many journals, and you will see more about that as we go forward.
A new generation of biohackers is tinkering with DNA to create plants with new colors. At the School of Visual Arts in Manhattan, plant researcher Sebastian Pachoco is trying to create genetically modified plants, mostly from information he learned on the internet. One of his pursuits in the school's bio-art lab? The blue rose, considered the holy grail of horticulture.
I think the main reason why people are trying to make blue plants is because there are very few, if not any, blue plants out there that are manipulatable or growable in a specific condition. Basically because those high saturation colors just simply do not exist in nature.
GMOs, or genetically modified organisms, have drawn a considerable amount of controversy, with concerns about the potential impact on health and the environment. Kachobo doesn't think such worries apply to projects like his.
The main argument I would have towards anybody who contests us going through this project is the fact that it will be contaminated into plants. So we're not working with pathogens or any other type of human or mammalian tissues.
Suzanne Anker sees the process as more of an art project than a science experiment.
Artists here use science as a tool, so they are not engaged in research in the same sense that a scientist would do. Blue is kind of an area of wonder. Children say, why is the sky blue? All the time. That's one of their first questions. And I think that is in part the background of this. But I do think that even if it doesn't look, it doesn't matter because it's art.
The process is meticulous and complex. It all comes down to identifying and manipulating a specific gene that could give the plant its color.
The gene in question, called AE blue, produces a beautiful cerulean hue in bacteria. It's never been tested in plants as far as I know. So this will be the first time that we actually try to put it into a plant.
With the project about halfway done, Kachobo is still tackling problems, but he says even if it fails, the experiment won't be a loss.
Okay, in an ideal situation, in another 3 months, it will have a replant. In the worst case, it might not work at all, and that's the risk that we're willing to take, and that's the risk that we want to attempt, because even a failure might be useful in terms of understanding the process better.
This was a piece that the Wall Street Journal did for us. It also appeared on the front page of the Wall Street Journal, which never happens to artists and art students. And my comment that said, if it doesn't work, it doesn't matter because it's art, is not a snide comment. It sounds snide, but it's not. Because the theory behind that is that for every invention or production in the scientific world, there is a distinct and unique cultural equivalence. And we'll see that in my work as we go forward. There's double identities in many of my pieces. So the question is, what is bioart? It's a contested territory. No one really knows who coined the phrase, although a lot of people take credit for it. And I have written the definition of it for the Oxford Dictionary of Art and divided it up into 3 subgenres. One is wetland, where you can see Mark Quinn's genomic portrait of Sir John Sulston, in which he took his cells and replicated them to make this portrait. So essentially, a viewer looking into this portrait will see themselves because of the stainless steel print. This is the first abstract portrait to be held at the Portrait Gallery in London. And this is about decoding DNA, which is why you see it in 2001. There are a lot of problems I have conceptually with the fact that this portrait is DNA, because DNA is not fixed. It is a substance that can change over environmental issues. The second subcategory has to do with software programs and the writing of DNA, as we see with CRISPR right now. As you've all experienced it, you've had the COVID vaccine. That could not have been possible without decoding the genome and then rewriting it using certain kinds of gene sequences and synthetic biology. And the third subgroup is any media whatsoever that deals with the issues of the scientific revolution that we're here now. Patricia Piccinini in the Young Family, she's an Australian artist, was in the Venice Biennale, and she created this image that appears to be part human and part God. And it raises a lot of philosophical questions concerning the fact that if we can do something, should we do it? And technology is essentially going faster than any kind of ethical kind of determination. We had the same question when we started talking about gorillas and other sentient creatures and whether they have certain rights and privileges because they're not just stuff. In 2009, I started to grow plants at Exit Art in New York City, and these were different kinds of plants that produce tomatoes and beans, etc. It's called Shelf Life because all of the components here were purchased at the Container Store. So essentially, I put together existing parts to create this, and I would go there on a daily basis and take photographs of the produce being grown. This is no Photoshop. This is red and blue lights that are part of this spectrum in which photosynthesis occurs. So you can really say that fuchsia is the new green. And part of the idea of this, and we'll see in a few slides later, is that agriculture is moving indoors because of climate change, because of the dissemination, rather the destruction of pollinators, etc., that certain kinds of foodstuffs in our food supply can no longer be in the places that it once was. And here you can see the little sprouts, until finally in 2015 I get a call from Herbie Rubin, who's doing the show at the Cathedral of St. John the Divine about the value of food, and asked me if I would like my own chapel. I said, okay, I had these 3 wall works, what do I do now? Well, I took the 3 boxes and turned them into 31 boxes, and the aura around the chapel was so pervasive that you could see it before you even came into the space. Now, people from all over the world come to this cathedral, and school children, visitors, etc., and they didn't believe it was real. They had never seen anything like this. So if you would essentially shine your light onto the vegetables, you would see that they were actually green and not fuchsia. This is the way a lot of your vegetation that is grown indoors, particularly ornamental plants, right now exists. And what was interesting about this was an article, why an artist is growing space lights. Okay, I used the same seeds that the astronauts used as they went up in space. And when this article kind of came out, it existed in 2 other places. One, it was in the video inside taxi cabs, and 2, an Italian salad magazine called me up to find out how to grow the space lights. And here you can see what the astronauts are doing. This is the Veggie 01 experiment at the International Space Station. You see the aura of fuchsia. And again, here's Scott Kelly, one of the first astronauts to eat the food in space. A little earlier than that, Japanese astronauts went up and they grew cucumbers, but they were not allowed to eat them because they were not sure whether there are any unknown kinds of microbes that might have affected the schools or the food supply. But what's interesting in outer space, where we're going at a fast rate, in astrobiology to be sure, is that botany doesn't function the same way. If you plant a seed on Earth, you get 1 sprout. If you plant a seed in outer space or anti-gravitational conditions, you get 2 sprouts. If you boil water on the Earth, you get a lot of little bubbles. If you boil water in space, you get one giant bubble. So everything we kind of know about botany on Earth could be revolutionized as we move on to other planets. And here you can see their Romaine lettuce grow. Another piece I'd like to talk to you about is my series Vanitas and Petri Dish and Remote Sensing. This installation first took place at the Parish Museum in Lauderdale, and it was based on the 18th century concept of Vanitas paintings. Now, Vanitas paintings came into existence because of a Dutch golden age in which the mercantile class was very wealthy. And it became a situation in which paintings contained time clocks or skeletons or decaying fruit, etc., to talk about the fact that everything has an expiration date. And what I did with Vanitas and the Petri Dish is bring it up to the 21st century. And we will look at that in a while. But it had a companion piece, again, based on NASA's satellite data, which was remote sensing from 2016. So for every two-dimensional image, there was a three-dimensional image that was created by a computer to turn a still life into a landscape. So this blurring of boundaries in our history now becomes an issue, and we have to think about what other boundaries are being broken. You could see the exterior here of these pieces that have been generated by machine learning, aka AI, in which the data that was read was a glitch and was something I had not conceived of, but was very intrigued and tried to repeat it. Here you can see them up close. They're very cute, by the way, and they do look like fantasy landscapes. Here are some of the Vanitas, which bring together objects from nature and objects of culture. Now, since the 1990s, we have been looking at the spectrum between the artificial and the natural. We now know that it is a spectrum, and that we eat plastic every day. So the rubber ducky is probably on an interplay along with the sunflower at some point. But synthetic biology can make a kind of a gene sequence that doesn't exist in the natural world, but they possibly cure disease or do other things. So we are in a revolution in the laboratory and outside of the laboratory. Here we see meat with some paper cutouts, some dead flowers, some shrimp with glass as ways to think about the mix and match of what is not only in our diet, but what is in the natural world. Now, using the 3D programs here, you can see that 2D turns into 3D in this way. A z-axis is added to the two-dimensional axis to create a form in 3 dimensions. This is called displacement mapping. Another show, and what I want to talk about here is how ideas grow. That just because you start a series and you think that it's finished, it may not be finished until you have to put it in another context. And in 2019, I was able to have an exhibition at the Kieferson Museum of Art called 1.5 degrees Celsius, which is the tipping point by which weather changes can destroy the earth. And here you see now everything is doubled in size. And what was interesting about this show is that the people who came to see it would try to match up the little remote sensing with the large-scale Vanitas, which created a sense of perceptual games because it wasn't always easy to see in both 2 and 3 dimensions. The glass vessels also, which I've used in other work, function as ways to store images. And these are just normal kinds of glass vessels on a Petri dish. Lo and behold, I think I'm finished with 48 Vanitas in a Petri dish until I get a call from the Beijing Biennial that want to work. Okay? So what they do is they want it bigger. So what we ended up with is over 75 Vanitas in Petri dishes, and here you can see them more. And finally, we go from the little show at the Parish Museum into the show called Synthetic Ecology in China. So an idea is never over. It just continues to take on new forms. And keep in mind that the opportunity to show in larger spaces creates bigger ideas sometimes. Another piece that I have worked on is this piece called Biota. And it taught me a great lesson because I knew I wanted to make these pieces. This is porcelain. This is a sea sponge that has been burnt out and is made of porcelain. Now, I knew this could happen, but I didn't know how. So first I took the sea sponge and I tried to scan it in one. I then tried to spray paint it to look terrible. I then tried to dye it. It didn't work. I then tried to cast it. It didn't work. And then finally, using this burnout technique in ceramics, the sea sponge evolved. So if I had given up, this never would have happened. So if you have an intuitive idea, if you want to make something, and it fails for 4 times, continue on. And I ended up making 150 of them. And again, this was part of the show in Syracuse. Each one is unique, and the sea sponge is a distant cousin of us. We share 70% of our genes with the sea sponge. And the sea sponge is a model organism because it has everything you need in order to make a nervous system, but it never made one. And so scientists are looking at those kinds of enzymes for possible cures for ALS or Alzheimer's, etc. The other thing about the double identity issue here is not only is it sea sponge, but it looks like dead coral. And 90% of our coral reefs are under attack at this point. So the coral reefs are like the Amazon jungle. They're the lungs of the ocean. And what is happening through pollution and global warming is that these reefs are being destroyed. Darwin was very interested in coral reefs, and this was part of the Darwinian paradox. He wanted to know why in a dead sea, where everything has no life in it, there springs up a coral reef which is fecund with all sorts of living creatures. And it turns out that the coral was in a symbiotic relationship with the photosynthetic algae that again fed the sponge and then fed some of the other creatures living. So this question is this a coral reef? Is this the brain? Is this a metaphor for the way in which neurology can be looked at? Because we've never looked at sea sponges. Sometimes they are like brain parts. They look like your brain. Here's a close-up of another installation using the biota pieces, and they have little figurines in them. And those are little rapid prototype sculptures put in because there are many theories that when primitive mankind started to create figurative sculpture, their brains changed. So here, again, the dead coral is a brain with little figurines. Silent Spring, Rachel Carson, 1963, had a great impact on the art world. Many artists read her book. The reason she called it Silent Spring is she was researching why robins' eggs were not hatching. And it turned out that DDT was causing the robin's eggs to be too thin to hatch chicks. She was ridiculed, she was brought in front of Congress, she had no PhD, she was a woman, and she was right. Okay? And I'm going to try to show you.
Ms. Carson maintains that the balance of nature is a major force in the survival of man, whereas the modern chemist, the modern biologist, the modern scientist believes that man is steadily controlling nature.
Now, to these people, apparently the balance of nature was something that was repealed as soon as man came on the scene. Or you might just as well assume that you could repeal the law of gravity. The balance of nature is built of a series of interrelationships between living things and between living things and their environment. You can't just step in with some brute force and change one thing without changing many others. Now, this doesn't mean, of course, that we must never interfere, that we must not attempt to tilt that balance of nature in our favor. But unless we do bring these chemicals under better control, we're certainly headed for disaster.
Well, you saw the children in the swimming pool and the hose with some chemicals being sprayed on them. Well, that was DDT. Okay? And DDT was also used on Japanese prisoners, American Japanese prisoners, to sort of clean them from any possible infections. So this issue of us in the environment, we know that in art making we have figured ground relationships. So if I was to sketch every one of you here, I would have a kind of portrait around your head. But you're in relationship to the air above you. That is part of you. But you don't think about that when you go about your daily life. Certainly in making a drawing, you realize that figured ground relationships are what makes something visible And in this case, it makes something alive. Which brings us to the current show. And I hope many of you have seen it. It's called Arctic Gardens and the Sixth Extinction. I'm not sure if you realize that, yes, that strawberries now grow in Greenland. And the famous Georgia peach, it's too hot in Georgia to grow the Georgia peach. And that the Arctic was once as warm as Italy. So these rotations of these cosmic forces in weather, which we're all experiencing now, because we know that one day it could be very warm outside, and by the evening it could be very cold. Recently, hailstones the size of baseballs were in Texas, raining down. So it's not only the Anthropocene, it's the mudslides, it's the Pliocene, and it's your experience that brought me to make this series of mine. This was not essentially an intellectual endeavor. It was a way I experienced the world outside during the pandemic. And everything in these pictures I grew, except the weather. And what I did was I brought more than one season into focus simultaneously. So there would be, as we can see here in the upper left, that there's fire and ice happening at the same time. And I think that that is an indication of how nature is now fractured and what can we do to now help alleviate that. In addition to these large-scale photographs, which are all collages, there is a series of rapid prototype sculptures as well, which are 3D variations on these. And here you can see them lined up in a row in which the perimeter looked like colorful paintings. And these are hand painted after they are printed so that they become archival, and they represent a landscape that is now under threat. Here you can see more closely the color in a sculpture which has always been a questionable premise. And in this little piece, the up and down structure of the perimeter is something I had not thought about, but the machine decided that that's what the data says, and I agree. Another piece here, Snow Petals, is also a rapid prototype sculpture, and this one is on the wall. And you can see in this slide here that the relief structure of the work adds another dimension to it, so it becomes both a painting and a sculpture simultaneously. I'm just going to show you some close-ups. Here is the snow petals. See, what I did is I went outside my house and I took all these photographs. And then after printing them out, I said, these are just too traditional. So I cut them all up. And in cutting them up, I began to see things that were color and form as opposed to representation. And that was a really important step. This one's my favorite. It's called pyrexia, and pyrexia means fever. And when the earth has a fever, we're all in shock. Arctic garden, snow, winter, and summer. Purple mirage, we see that the purple in the center piece is fading away, because that's an invasive plant. Hybridization in red gives you a complex series of ferns that don't exist in nature, so they may have been made in a lab or mutated under other conditions. Split spore. This brings into question reproduction. Now, mushrooms reproduce by spores, so do your dandelions. But what about our seeds? Who owns our seeds? You know that seed companies are all owned by chemical companies now? After World War II, they bought up all of the seeds. So the food supply now is in the hands of the same people who use Agent. And lastly, I'm going to show you a project I did in 2016 with my students. Because I don't consider my art practice any different from my teaching practice, that it is all one yarn. And in 2016, way before Jan Berger or Impossible Burger, we did Mayo tomatoes. And what we did was we generated a tomato by genetic engineering that had the same protein as a steak. So you can see in this poster that we eliminated the cow and created a tomato that could be grown by any poor farmer anywhere. We did a culture in vitro. And we presented this at the Museum of Modern Art in the Biodesign Chapel. We did win an award. But I want to tell you a little story about this tomato, which looks a lot like the Aztec calendar. Because the tomato is from Central America and Mexico. And then, because of colonialism, it ended up in Europe. It did not come right up to the United States. So it had to go across continents in order to come back. And every part of the tomato plant is poisonous except for the tomato. So don't eat any roots or leaves. This was something that many Americans were afraid of. There's also other stories about whether a tomato is a fruit or a vegetable. It's really a fruit, but because of carrots, we call it a vegetable. This was our scientific process in which we did protein research, we worked with scientists in the lab, and it's a pun on the beefsteak tomato. And then finally, using the gel electrophoresis, it indicated that the myoglobin gene, which is like hemoglobin in humans, had been integrated in the tomato genome. So we actually put this protein in the tomato. We didn't know where it was, we didn't know how much it was, because we don't have PhD students here. We also created a bunch of products, like the Mayo tomato soup, bringing it back to art, the diced tomatoes, the ketchup, etc.
Hi there. Welcome to the SBA BioArt Lab, where we've been designing the future of food for over 30 years. Good. This laboratory, the first of its kind, is where biologists and artists work together tackling some of the world's biggest biological concerns. Over the years, we've spawned some unique results, but nothing as successful, impactful, and delicious as this. Meet the Mayo tomato, the amazing protein-packed fruit that started it all, a biological breakthrough that changed the way we think about protein. I have something amazing to show you. Randy, get the thing.
As you wish.
Before the biotomato, humans relied heavily on animals for their daily source of protein. With a growing global population and a strained agricultural system, we recognized animal protein was not an ecologically sustainable source. There had to be an alternative method. We put together some of the brightest scientific and artistic minds we could find, and the result was impressive. Take a look.
This little man a little bit. JK.
Oh.
2016, the BioArt team at the School of Visual Arts engineered the world's first fruit to produce the same protein, myoglobin, found in animals, the myotomato. They found plants to be deficient in megadoses of protein necessary for a nutritional human diet. So they translated a DNA sequence found in beef and inserted it into a tomato's genome. The result? One little tomato possessed the same amount of protein found in an 8-ounce steak. This breakthrough creation caused quite the stir. It seems not everyone was a fan of this amazing scientific achievement.
GMOs are dumb and stupid and poison.
Of course, we're not happy.
However, in the year 2030, mad cow disease almost completely wiped out the cattle industry, creating an urgent need for the myotomato's amazing protein powder. And soon, hearts, minds, and mouths were open. The myotomato ushered in a global movement that ultimately ended our need for animal consumption. Not bad for a group of art students grasping at the moment.
This project has traveled many places, including Harvard and MIT, et cetera, et cetera. We've done another project as well, but we don't have time to talk about that. But for those of you interested in following any more of this discourse, here are some of the books that I've written, just a few, and here are some more, and I just didn't want to bore you with reading. So, thank you very much. Any questions, anybody? I have a lot of very smart people here, so don't ask me too hard. We couldn't bring it to market because we didn't have a license to do so. When you're meaning to commercialize as a product, you would have to go through all sorts of red tape. We're not PhDs, and we would have to really figure out how much protein and where it was in the tomato in order to sell it. So we did take it a little further, but then it became outside of our realm. So we consider it a speculative project, and I would like to use that word to everyone here, because if you can think about it, eventually it will be possible. And speculation, very important.
I just wanted to know, how did the tomato taste?
We didn't eat it. Anything in the lab is not food, okay? So, I don't know what some of my assistants may have done, but I have never tasted it, okay? We do have the seeds, and we do have a kind of freeze-dried version of it, if anyone would like to try it off campus. It would be a possibility.
I'm interested in this correlation, the observations made between nature as fashion and your use of fashion. Change it back to one space.
Well, thank you. I think this is true, and this is true. As I said earlier in the talk, for every scientific proposition, there is a cultural equivalent. It might be distinctly different. But even in the first clip I showed you about the blue flower, that comes from Novalis, the 18th century philosopher poet, who was in love with a 12-year-old girl, and she died, and the blue flower became a symbol of the ethereal, of the infinite, of all of that is untenable. Now that symbol still exists in film. David Lynch has used it, and Stanley Kubrick has used it in Eyes Wide Shut. So there is the symbolic value of what becomes a sort of scientific hypothesis. Now, have scientists been able to make a blue rose? No. Okay? One of the reasons is that the pH of blue in horticulture is very unstable. But if anyone can make a blue rose, there'll be a billion. I did buy some blue rose seeds on Amazon to see what would happen. No. And anytime you see a blue rose in the store, it's dyed. It's put into blue liquid, and to the vascular system of the rose, it turns blue or it's pregnant.
Thanks. Yeah, still a fully formed question. I'll take it back. Was there a point at which, when you were a young artist starting out, where your practice became activated by all this? Your practice remains activated, but there's also, to me, like a layer of, I don't mean activism as in marching in the streets, but it's an active and activist practice because of the forward momentum of it, if you call it. And so was there a moment at which, in your education, that happened? Was there a breakthrough or an insight? And can you talk a little bit about it?
That's a great question, and I'm going to answer it with two anecdotes. The first one is, I grew up in a Fred Trump building, okay, in Brooklyn. Okay, and they had lawns and trees in front, but I didn't like any of that, so I went to the empty lot behind my apartment building, where I collected milkweed and caterpillars, brought them home, and grew butterflies. And that sense of wonder stayed with me, that this idea of metamorphosis and the wild was my inspiration forever. It still is. I think that the other part of activism is all art takes a proposition, okay? It may not be overt, and certainly that in this techno-scientific culture that we live in, we have to now demand our rights and privileges as living beings. My earlier work was involved in genetics, what happened is I had a show in New York, and I had kaleidoscopes on some of my sculptures, and when I looked at this bowl of eggs, it looked like a cell, a fetus, and all of these other images. So I went home and took out my daughter's biology book and looked for something that was not a disease. And I came across the chromosome and studied that very fervently, and decided that chromosomes were the way the body writes itself. And then in 2004, with the Sociologist of Science, I wrote the first platform called The Molecular Gaze, Art in the Genetic Age, published by Cold Spring Harbor Laboratory Press, where I met Watson, etc. Okay? So it was through the magic of the invisible that drew me to where I am now. And I'm still overwhelmed.
Do you think that edits toward food's genome for reasons other than vanity and the human fixation on perfection could be translated to the larger socioeconomic issues that we face globally as the hunger crisis extends to much of our human population? And would there be a larger experiment calling for food modified to create a balanced meal supply?
Well, I think that food shortages and food deserts, which you find in certain economic zip codes, as well as in different countries around the world, some GMOs are not all bad, okay? But we'd like to know what's in them, and there's no label in the U.S. There are ways to modify crops so that they don't get viruses, for example. But again, back to eating the seeds and who owns the seeds, this is a real problem. Because farmers used to, after their crop was finished, collect the seeds and then plant them next year. But now the seeds have a terminator end to them, so they cannot be replanted. So you have to go buy them again, probably from Monsanto. It's not the pesticide on it, but the pesticide in it. So this is really a question that requires a lot of thought. We do have a course here called Food in the Era of Climate Change. It doesn't get much takers, but I highly suggest it because it's very, very interesting. Anybody else?
Just a little curious to ask you, like, do you enjoy more to work with technology and science or fine art and photography?
My background is not in science, although I started out in college studying science, but I went out on too many dates, so I didn't do well on the exams. And that's when I switched to art. And my teacher was at Drawing Project. And he introduced me to the idea that art was not only for dumb people, but that art was also about philosophy. And that changed my life. I think we'll wrap it up. Thank you all.